Hi again, it's me, Kristen, and I wanted to tell you today that Sarah and I couldn't do all of these videos and these programs without help from some of our partners. And today we have a partner here with us named Joe Deerdorf, who is going to talk you through your plant diversity lesson.
Hi everybody, and welcome to the plant diversity section of our virtual Salmon Watch. My name is Joe Deerdorf and I'm the botanist for the City of Albany, and today we're here at Bryant Park in Albany, Oregon in a healthy riparian forest. So, have you ever heard of a botanist before? What do you think a botanist does? Botany is the study of plants, so a botanist is a person who studies plants professionally. Sometimes we may be talking about agricultural plants or garden plants, but in this case, we're going to be talking about forest plants. Okay. Now we're going to do an activity. You're going to go to a window, look out from a door, or go outside. We're going to spend 1 minute observing the different plants that we can see there. Pause the video while you do this and resume in 1 minute.
Welcome back. Did you see any plants? How many total plants did you see? Remember, every individual organism counts as one plant.
1, 2, 3.
Did you see many total plants, or did you see only a few total plants? Now, how many different species of plants did you see? Did you see many species of plants? Did you see only a few species of plants? Or did you see only one species of plant?
1.
These are the kinds of observations botanists make. We determine how many plants are in an area and how much diversity there is in a plant community. Diversity means the number of different species of plants within a plant community. If there are a lot of different species of plants in an area, we would say that that is a diverse plant community.
Here's a photo of a conventional lawn. Notice that there are many total plants. Every blade of grass is an individual, but there are very few kinds or species of plants present. In this second photo, you can see a meadowscaped lawn. The site is very similar, but you can see flowers, trees, and other plants. Which is more diverse? Clearly, the second habitat has more diversity because there are many different kinds of plants. Before we go into the field, we're going to build an important botanical implement called a plant press. A plant press is an important tool we use to keep a record of the plants that are present at a specific location. Build your plant press using the following steps. First, gather all of your materials. 2 pieces of blotting paper, 2 pieces of cardboard, 2 rubber bands, a pencil, and a heavy book. Trace the cardboard on your blotting sheets. Cut out or fold and tear the traced cardboard shape on your blotting sheets. Your blotting paper should be the same size as your cardboard sheet. Place the cut sheets between the cardboard pieces to make a cardboard sandwich. When you're done, the cardboard should be on the outside. Wrap the rubber bands around the top and the bottom of your cardboard to keep everything in place. Add your name to the top of the cardboard piece and decorate it if you want. And then we'll use our book to press our plant press down with the plant inside of it later. Your finished product should look something like this. We're just about ready to go collect our samples in the field. Before you go out to collect your samples, make sure that you have your plant press, your plant diversity lesson, your data sheet, your plant ID key, your plant guide, and a writing utensil. One note before you go out, we want to be really careful of poison hemlock and poison oak, and you can find pictures of these in your lesson. So once you have your plant press ready, you're ready to go collect your samples. Now it's really important that you go somewhere that's on your own property or at like a public park or at a friend's house, and you don't go onto someone else's property to collect these samples. I really like Oregon grapes, so this is the plant that I'm going to collect. So when you're collecting your samples, you don't want to pull the whole plant out of the ground. You just want a small enough section that you know it's going to fit into your plant press. Okay, so actually this is too small of a sample for our plant ID because we actually need the bud included. So when you take this sample, you want. So what I have here is two entirely different leaves with pairs of leaflets, and each leaf has a bud on it like this. Okay, so now that we have a compound leaf, I'm going to show you a simple leaf. And we have a 9-bark here that we'll take our specimen from. Okay, so now we have a simple leaf compared to the compound leaf that we pulled earlier. The difference is that this is one single leaf here, whereas this is several leaflets that make up one leaf. And you want to make sure that you've got the bud on your sample as well. Okay, so now we're going to use our plant identification key to identify the two specimens we collected. For this video, I'm just going to demonstrate the one, and we'll do this one with the leaflets here first. So number one on here is, what kind of a leaf is it? Is it a broadleaf, a needle, or does it have scales? And so we can see that this has leaves, so we would go to number 4. And that's on the back page. So is it simple or compound? And so you can use this hint right here. You want to look for a bud that tells you that it's one single leaf. And so if any of these were a leaf, you would find a leaf bud at the base of them. But for this one, there's just one really big bud right here. And so we know that all of these are leaflets and not leaves. So this one is compound. So we'll go to number 12. Okay, so we'll move on to number 12. Are the compound leaves opposite or alternate? And this one can be a little bit confusing for compound leaves, because remember, each of these are leaflets. They're not actually leaves, and so the leaflets are on opposite sides from each other. But we'll have to look at the larger plant and not our specimen to be able to tell if the leaves themselves are opposite. And so I grabbed another larger sample so we can see that the leaves themselves are alternating. If they were opposite, then they would be coming off from the same point on the plant, but instead what we have are alternate leaves. So we'll go to number 17. Okay, we're on to number 17. Are the leaflets lobed and feathery, pinnately pointed, or palmately compound? And so you can see the example of Himalayan blackberry here is palmately compound, and that means that the leaflets form a shape that kind of looks like your hand. You can see the shape that this makes there. We know that that's not what we're looking at here. Our next option is pinnately pointed, and so if the leaflets are pinnately pointed, then they're kind of spear shaped, which is what our sample looks like, and you can probably tell what this is already. But let's just check the last one just to be safe. Lobed and feathery looking, which you can't see maybe in the video, but it is definitely not feathery, it's really pokey. So we know that this is Oregon grape. And once you've identified your plant, you're going to go over to the Oregon Plant Guide and look up the species that you identified, and you're going to write down some fun facts about that plant.
Right on the data.
Okay, now that we're back home with our plant samples, we can go ahead and press them with the plant press we made earlier. So just make sure that all the leaves fit in the area of your plant press. If you need to, you can make them a little bit smaller. You just want to make sure that you still have the little bud on your sample. So there's one. And this one fits just fine in between. And then you're going to stack the samples on top of each other and sandwich them all with your other piece of cardboard. And then wrap the rubber bands around the top and bottom edges. And then once you've got them inside your plant press, you're going to put your big heavy book on top of it, and you're good. After you have collected your plant samples, you will move on to collecting plant diversity data. For this part, you will need your data sheet and a writing utensil. To collect plant diversity, you will pick 2 or more different locations to collect data. This can take place at 2 different areas of a park, your neighborhood, or any area where you can count plants. At each location, you will stand in one spot and record how many different species of plants you can see. Make sure to look high and low, as well as turning around in a circle to count all the plant species that you can see. Once you have finished counting, record the number of plant species on your data sheet. Then move on to your next location and repeat these steps. Let's look at the data sheet. First, you're going to fill in the location. For me, I'm at Bryant Park. And then you're going to fill in the number of plant species that you can see. In this location, I can see 25 kinds of plants, so I'll fill that in on my data sheet. A helpful hint, if it's wintertime, when you're looking at these plants, is to look at the ground underneath different plants and see what different shaped leaves you can find, and that'll tell you if they are different species of plants.
Now that we have 2 plants identified and pressed, let's go back to the data sheet. Remember how we recorded the number of different species we saw at each site? Why do you think we did that? We did that to get an idea of the diversity of the different sites. So why do we talk about diversity? We study diversity to know more about the biodiversity of a region. Biodiversity is a combination of bio, which means living things, and diversity, which we talked about earlier, is different types of organisms in a community. All organisms make up biodiversity, so that includes plants as well as fungi and animals like mammals and fish. So having a really diverse plant community is really important for improving biodiversity by providing shelter and food for animals that live there. Having a healthy, diverse riparian habitat like the one I'm standing in here at Bryant Park is important for salmon and steelhead, and you're going to learn about that in the next video. That's it for me. I hope you all have a great rest of your virtual salmon watch. Bye-bye. 98,991, 98,992, 98,993, 98,994,